Hey guys, this is 희민이는 from Elena to share.
The theme of today's video will be protein. This is one of the concepts that I think are most important in this unit, so please be focused. During my previous videos, we got to know that carbohydrates and lipids consist of carbon, hydrogen, and oxygen. However, today's macromolecule protein is made up of carbon, hydrogen, oxygen, and the special one, nitrogen. So what does protein do in our body? As you can see, there are so many roles of proteins. Enzyme structural proteins, contractile proteins, defensive proteins, signal proteins, receptor proteins, transport proteins, and storage proteins. So let's quickly take a look on each one of them. First one is enzymatic protein. Among many types of them, the most familiar one to you guys would be digestive protein. If we eat some food, those digestive enzymes help breaking down those foods to smaller bits. And the next one, storage protein, functions as storage of amino acids. And defensive proteins are the ones who defend our body against diseases. We'll look closely on transport proteins in Unit 2, but in short, those are the ones who transport substances. Do you remember when we talked about iron deficiency? It was on the first video. Iron is important to living things in that it makes hemoglobin. Hemoglobin is the most familiar example of transport proteins as it transports oxygen. Also, protein forms hormones. Then you might ask, how about lipid? We learned in the last video that lipid forms steroid hormone. That's right, but steroid hormones is the special one. All the hormones except steroid hormone consist of proteins. Familiar proteins-based hormones would be growth hormone and insulin. Moving on to the receptor proteins, they receive signal from nerve cells. So its another name is signal proteins. Contractile and motor proteins are actin and myosin that consist of our muscles. And lastly, structural proteins form hair, horns, feathers, skins, and webs of living things. So, in macromolecules, there are monomers except lipid, as I said in my last video. Then there must be monomer of protein, and that is amino acid. There are 8 essential amino acids, and polymer of them is polypeptide. The picture below shows the structure of amino acid. Each of the group, like amino group and carboxyl group, is called functional groups. Amino group is NH2, and carboxyl group is COOH. However, the most important one is R. It is side chain and called R group. This R stands for random, and it decides the property and what amino acid it would be among 20 types. So the structure of amino acids depends on R group, and everything else still same. So let's take a look at those 20 types of amino acids. The box with purple color are the common groups, amino and carboxyl group, and yellow ones are the random groups. Previously I mentioned that CH is nonpolar, and you will see at the top that it says nonpolar hydrophobic. So easy way to memorize this is if amino acid consists of CH or ring thingy like tryptophan or phenylalanine, it is nonpolar amino acid with very high possibility. There is also the smallest amino acid, glycine, in the top left corner. So this was nonpolar one, and there must be polar one too. The polar side chains are hydrophilic, and similarly, purple ones are common parts and green ones are the different parts. Easy method to memorize them is just knowing that they have oxygen, hydrogen, or sulfur. But then some might go like, but there was sulfur in nonpolar one too. If you look carefully in methionine, there are far more CH than S. And also someone can go like, there was nitrogen in nonpolar side chains too. That's right, but if you just remember that one, that nitrogen, it has nitrogen but has rings, like tryptophan is nonpolar. Okay, moving on to the next. Last one is electrically charged side chains, and they are also hydrophilic. It is separated to acidic and basic ones. As acidic chains are negatively charged, if there is any negative sign, you will recognize that if it is acidic and electrically charged side chains. And if you see plus sign, you'll recognize that it is basic and is positively charged. In exams, you don't have to memorize all the name of the amino acids, but you should be able to tell whether they are nonpolar, polar, or charged when structure is given. The bond in protein is peptide bond, and dehydration synthesis occurs. I'll emphasize again that name of bonding are very important. This is the most important thing that we will study today, so listen carefully. There are 4 levels of protein structure, and each of them has different functions. Primary structure is just simple connected amino acids. When protein changes to higher stage, it is folded once. There are 2 kinds of secondary structure, alpha helix and beta pleated sheet. There are hydrogen bonding between amino group or carboxyl group of adjacent amino acids. Proteins consist of either alpha helix, beta pleated sheet, or both of them. In these 2 stages, the protein is not able to function yet. So for it to function, it at least should be tertiary structure. Tertiary structure is 3D shape, and it does function. It is made up by interaction of R groups by hydrogen ionic or covalent bonding. Quaternary structure is made when 2 or more peptide chains are connected. So the difference between tertiary and quaternary is that tertiary structure functions alone, so it is a single unit that functions. However, quaternary structure is a group that functions together. Great example of quaternary function would be hemoglobin. It has 4 peptide chains as shown in the picture on the right. Finally done with 4 levels of protein structure. Now we move on to chaperonin. Chaperonin is a protein that assists in proper folding of proteins. I said that folding changes level of proteins, which is very important procedure. It consists of 2 parts, cap and cylinder. For it to function, the cap opens first, and unfolded polypeptide enters the cylinder. Then the cap attaches, causing the cylinder to change shape in such a way that it creates a hydrophilic environment for the folding of the polypeptide. And finally, the cap gets off, and the folded protein is released. Proteins have important property that they are very sensitive to chemical and physical conditions. pH or Temperature must be an optimal condition for bonding. If the condition is not optimal, it is not able to do bonding, and so not able to function. This was the last thing I was going to say in this lesson. Today I talked about very important concepts in first unit, and I hope you guys followed well. Please like and subscribe if this video was helpful, and I'll see you in my next video.